So remember how we said that the whole point of eating is so that you could get all these nutrients and then digest them and catabolize them. So the whole purpose of all of this is to get some energy, because we want to get energy out of the food we just ate. All right. So we want to know, how do we get energy out of glucose? And it's not just a one-shot deal. It's several processes that have to occur in order for you to get your energy. Now, I don't know if you remember the word mole, but 1 mole of glucose gives you 30 moles of ATP. Okay? If you remember from intro chemistry, a mole is 6.02 times 10 to the 23rd. To simplify, we're just going to say 1 glucose gives you 30 ATP. So we have to find, how in the world are we going to get 30 ATP from 1 glucose, or 30 moles of ATP from 1 mole of glucose? All right, so this is what happens. Remember, in the previous video, I said glucose is in the cell, the insulin bound to the receptor, the glucose is in the cell, it's in the cytoplasm. The first series of events are those 10 enzymes that occur in glycolysis. So what I have here is glycolysis. Input in glycolysis, we have glucose, okay? You're going to actually input a couple of ATPs, but then we're going to get a couple out. So I'm not going to worry about the input of ATPs. I'm going to just focus on the output. So the main thing input is glucose. What is the output? After 10 enzymatic reactions, what do you need to know as the output of glycolysis? Now, if this was a full biochemistry class that was 16 weeks and 4 credits, you would learn every single enzymatic reaction, as they do in the other, like the Chem 1060 class. But for you guys, we're just going to focus on what we're starting with and what we're ending with. All right, so what are we ending with? The output of glycolysis are 2 molecules called pyruvate. These are 3 carbons. Glucose is 6 carbons. These are 2 3-carbon molecules. Just know the name pyruvate. You get 2 molecules of pyruvate, 2 molecules of ATP, and 2 molecules of NADH, H+. Okay, do you guys remember NAD? Remember how I told you in the last series of videos that NAD is an important coenzyme? And remember how I told you it's usually involved in redox reactions, and it can get reduced, it can pick up hydrogens. So this is super important to remember that, because we're going to be using these NADHs to get a whole bunch of ATPs at the end, all right? So what do you guys need to know? Input of glycolysis is glucose. Output is 2 pyruvates, 2 ATPs, and 2 NADHs, okay? This is all, where does it occur? It occurs in the cytoplasm. Okay, remember we said that. This is all in the cytoplasm. Okay, now the 2 pyruvates, they go over to the transition reaction. The transition reaction occurs in the matrix of the mitochondria. If you remember from biology class, your mitochondria is a double membrane. I'll move it so you can see what I'm doing here. There's an outer membrane, and then there's an inner membrane. And then this part in the middle here, this is called the matrix. All right, so the transition reaction, this is in the matrix of the mitochondria. It takes up so much space, but you get the idea. All right, so this reaction, what happens? Your pyruvates are turned into something called acetyl-CoA. Remember, CoA is an important coenzyme that we talked about that's derived from vitamin B5. Remember pantothenic acid? All right, so in this reaction, the net, the output, what do you need to know in the transition? You get 2 acetyl-CoAs, you get 2 carbon dioxides, and 2 NADH H+, all right? You don't have any carbon dioxides produced in glycolysis because we account for all the carbons. This is 6 carbons. Each one of these is 3 carbons. So we have 2 3-carbon ones. You accounted for all the carbons. Pyruvate is 3 carbons, but acetyl is only 2 carbons. So you give off 2 CO2s, 2 carbon dioxide. Every time you breathe out, just think this is where you're getting it from. Okay? A lot of the metabolic reactions, byproduct is carbon dioxide, and that's what you're breathing out. All right, so what are the products of the transition reaction? 2 acetyl-CoAs, 2 carbon dioxides, 2 NADHs. Again, remember, these guys are super important. We are going to use them in the electron transport chain. All right, and then we go to the Krebs cycle. Krebs is a cycle that has 8 enzymes. But it's called a cycle, not a pathway. Like if you look at your book, they've got a nice picture of the Krebs cycle. You do not need to know all those details. Don't worry about that. All you need to know is what you're putting in and what net you're getting out. Okay, so what are we putting into the Krebs cycle? You're putting in 2 acetyl-CoAs. This is 2 carbons and it's hooked up to coenzyme A. Again, don't worry about the structure, but that's what this is. This is an acetyl group, is just 2 carbons hooked up to a coenzyme. All right, and you're putting 2 of them in. Why are you putting 2 of them in? Because we had 2 of them in the transition. Why did we get 2 acetyl-CoAs? Because we had 2 pyruvates. Why did we get 2 pyruvates? Because 2 pyruvates came from glycolysis. So this is first, this is next, this is third. Okay, what else are we getting out of Krebs? So you're getting another 2 carbon dioxides. Actually, I take that back. You're getting a total of 4 carbon dioxides. Take that back. So 4 carbon dioxide. So total net, you have 6 CO2s are being produced from 1 glucose. So every single carbon of glucose is pretty much accounted for in the carbon dioxides. All right. So we got 2 carbon dioxides in the transition reaction, 4 carbon dioxides in Krebs cycle. What else are we getting in the Krebs cycle? And these are the important ones. 2 FADH2s. Do you remember FAD? We talked about it in the enzyme chapter. Flavin adenine dinucleotide. It's a coenzyme derived from, remember, riboflavin, vitamin B2, right? And the NAD was derived from niacin, vitamin B3. Remember that? Remember, that's why you're seeing all these B vitamins involved now in metabolism. So this is important. The NADH is also produced in Krebs, and you're getting a nice little bonus. You're getting 2 ATPs. They're actually produced as GTPs, but then we can turn them into ATPs. But anyway, what do you guys need to know? Because that's what you want to know for a quiz or a test. You are out of glycolysis. You're getting 2 pyruvates, 2 ATPs, 2 NADHs. Out of transition, you're getting 2 acetyl-CoAs, 2 CO2s, 2 NADHs. Out of the Krebs cycle, you're getting 4 CO2s, 2 FADH2s, 6 NADHs, and 2 ATPs. It seems like a lot to remember. Honestly, it's so much easier when you know the full story and every single enzymatic reaction. But I think that'll be too much for you guys to deal with. So just know the overview here. All right. So right now, net, what do we have? Oh, and Krebs cycle is also in the matrix. All right. So Krebs cycle in the matrix, transition in the matrix, glycolysis in the cytoplasm. All right. So right now, what we've got a total of, okay, so we're going to add the site. I wish this eraser worked better, because it's definitely easier than the, oh, it's not so bad. Okay. Yeah, that's not so bad. All right. So now we have the net output. What do we have? We have a total of 4 ATPs net. Remember how I told you that one glucose is supposed to give you 30 ATPs? Do you guys remember that? Are we anywhere near 30? We're not. We've only produced 4 ATPs. So that means the rest, all the 26 other ATPs, they're all going to be produced in the electron transport chain. Okay? How are we going to produce them? That's where all those NADHs and FADH2s come in. All right? So we're going to write down how many NADHs did we produce? What is the total number of NADHs? We had 2 in the cytoplasm, 2 over here in transition, and 6 in the Krebs cycle. So a total of 6 and 2 and 2, a total of 10 NADHs. The NADHs that are produced in the cytoplasm actually behave more like the FADH2s, but we don't have to get into the detail of all of that. But we'll put down over here that we've got 2 FADH2s. But technically speaking, like I said, those NADHs in the cytoplasm look more like the FADH2s. Anyway, these guys here, this bunch, this is what's going to help us get the 26 ATPs that we need to get. So in the next video, I'll do a quick summary of the electron transport chain, and then we'll leave this and we'll move on into lipids. Again, I could spend 10 videos talking about every detail of all of this, but that's not my intention for this class. So we'll just do an overview of the electron transport chain, show you how you get the ATPs, and then move on to lipids. Okay? Good.